Next part in the history of microbiology is development of microbiology in 19th century. This particular period is called as golden era of microbiology, which is from 1857 to 1954. This is called as golden era because the major developments and discoveries, inventions, significant inventions in the field of microbiology that took place in this particular period or era. So, due to the significant developments that took place in this particular era, it is called as golden era of microbiology. So you need to remember this period, that 1857 to 1954. One of the developments, significant development that took in the field of microbiology was related to the discovery of role of microorganisms in transformation of organic matter. In this case, organic matter is considered as food, and the spoilage of food or the making of different type of foods is a result of microbial activity. That is the principle behind this transformation of organic matter. That means the food is modified, and that modification is caused by microorganisms. So this is called as germ theory of fermentation. So it was proposed that the spoilage of food, it is caused because of microorganisms, and the change that occurs in transformation of organic matter, it is considered as fermentation. So this germ theory of fermentation, it states that the fermentation process means the transformation of organic matter. This process is caused due to microorganisms. That means microorganisms are involved in the transformation of organic matter, or microorganisms are involved in fermentation. So these chemical changes that occur in food or organic matter are fermentation and putrefaction. Fermentation means it is the process which is mediated by microorganisms that results in the formation of alcohols and organic acids, whereas putrefaction is a process of decomposition of results that results in the formation of ill smelling products, occasionally in meat, as a consequence of degradation of proteins. So these two changes are responsible for the spoilage of food. That was the theory, and it was caused by microorganisms. So the work related to this fermentation process as a biological one, it was first discovered and presented by three scientists in 1837. These three scientists were Latour, Schwann, and Kutzing. They were working on the fermentation by yeast. So they observed, or they proposed, that these are the yeast cells, means the microorganisms, which are responsible for conversion of sugar to ethyl alcohol and carbon dioxide. So that was the first theory which was proposed by these three scientists after studying on yeast cells responsible for the production of ethyl alcohol and CO2. Later on, it was Louis Pasteur who worked on the germ theory of fermentation. Louis Pasteur in 1856-1857, he worked on several fermentations, One of the fermentations he was working on was ethanol fermentation. During his study, he has made several theories and discoveries. That time, there was a French wine industry, and that French wine industry, it was facing a severe problem of production of a very bad quality sour taste wine. So this company was facing a huge loss because of the production of bad quality wine. That time, this French wine industry, they called Louis Pasteur to solve this particular problem because they were aware of the experiments or the work of Louis Pasteur. So they called on Louis Pasteur to solve this particular problem of production of bad quality wine. Louis Pasteur carefully examined that wine under microscope. At that time, good quality microscopes were discovered. So, using this microscope, he observed presence of bacteria in wine. These bacteria he identified as lactic acid bacteria. That means these bacteria were producers of lactic acid, which is an organic acid. So he concluded that these lactic acid bacteria are responsible for production of lactic acid, and this lactic acid turns the taste of this wine sour. So lactic acid was responsible for the sour taste of wine. After finding out the cause behind the production of sour wine, in order to now the solution. So in order to kill these bacteria from wine, he designed a process of gentle heating. So after Pasteur found the presence of contaminating bacteria in grape mash, it is a principal raw material for the production of wine. What he did, he gave a temperature treatment of 60 degrees Celsius to for 30 minutes to grape mash. And this heating process, it killed the lactic acid bacteria. Then this grape mash, it was inoculated with pure culture. Also, Pasteur gave this heating treatment to wine for its preservation or long period time. After this, the quality of wine was improved. Thus, Pasteur became a national hero after developing this technique for the control of undesirable microbes in wine. This process is now called as pasteurization in the honor of its inventor Louis Pasteur. Nowadays, this technique is used to preserve the milk and milk products in dairy industry. For milk preservation, pasteurization, temperature is 145 degree Fahrenheit, that is 63 degrees Celsius for 30 minutes, or 161 degree Fahrenheit, that is 71.7 degrees Celsius for 15 to 30 seconds. Pasteurization of milk only kills pathogenic microorganisms present in milk. That should be noted. But all microorganisms are not killed. Therefore, pasteurization is a gentle heating treatment which involves partial sterilization only. That means there is no complete killing of all the microorganisms present in milk. So it is called as partial sterilization process. Pasteur then worked on fermentation processes like lactic acid fermentation, alcoholic fermentation, butyric acid fermentations. And during his investigations, he discovered some very interesting points. First, Each type of fermentation is the result of microbial activity. That means in every case of fermentation, there is involvement of microorganisms. Second, during the fermentation, microbes produce specific type of end products using substrates like sugars. For example, in case of lactic acid fermentation, lactic acid bacteria are the microbes that converts lactose into lactic acid. Or in case of ethyl alcohol fermentation, yeast microbes convert that sugar into alcohol and CO2. For fermentation, microorganisms require a specific environment. For example, specific environment involves presence or absence of oxygen, then specific pH and specific type of temperature and raw materials. These findings are very important and very important in relation to fermentation technology. Louis Pasteur also has a contribution of discovering anaerobic life. Anaerobic means absence of oxygen. So microorganisms can grow in absence of oxygen. It was first discovered by Louis Pasteur. So discovery of anaerobic life. Pasteur published several research papers on fermentation during his study. He continued his study on fermentation for approximately 20 years, and during his study on butyric acid fermentation, he discovered anaerobic life in microbes, because in this process the microbes were unable to survive in presence of air. Pasteur coined the term aerobic and anaerobic. Aerobic means those which require presence of oxygen for the growth, and anaerobic means those which do not require presence of oxygen for growth. Another contribution of Louis Pasteur in the field of microbiology is again related to fermentation. He proposed the physiological significance of fermentation. Physiological means the function, why microorganisms carry out fermentation. The reason was given by Louis Pasteur. So physiological significance of fermentation. During his study on fermentation, Pasteur described the physiological significance. He described the fermentation process as the energy yielding metabolism. That means why microorganisms produce lactic acid, why microorganisms produce ethanol or other organic acid. The reason behind that, this production process, it yields or it generates energy. So this fermentation is the energy yielding mechanism for the fermenting microorganisms. He also stated that fermentation is a less energy yielding metabolic process than aerobic respiration.